So introduction to neuroscience, it's kind of a big field. We've been collecting facts like mad for over a century. And it's a kind of strange field because we know basically nothing. We have no clue whatsoever about the brain, how the brain works. The answer to all interesting questions about the brain is we have no idea. But there's a reason for that. And I want to sort of start off by talking about a little bit about why, and then we'll talk about the actual neuroscience facts, and then we'll go back to how we might understand, eventually make some progress understanding how the brain works. So I'm actually going to start about talking about science in general. So I don't know if you guys ever thought about what scientists do. I started thinking about this a couple of years ago, and the question was, what's it mean to understand the brain? And then what's it mean to understand anything? And it turns out in science, we have a very clear meaning for that. It's always, pretty much any science is always the same. We explain behavior in terms of equations.
What does explain?
So, I'm putting this in. We'll sort of see what this is. And these are broadly defined.
Yeah, Yasser wants to know what explain means.
This is that bad part about when your postdocs leave, you can no longer fire them. Actually, it means, that's a really good question. So explain is some kind of logical link in the broadest sense. But in practice, it usually means we write down some equations and solve them, if we're lucky. Okay. Now, we don't necessarily know where the equations came from, but the equations explain the behavior, then we declare victory and go on and look at a different problem. So I'm going to give two examples. So the first one is kind of easy. So you're sitting on a cliff and you take a ball and you drop it, falls, it hits, you drop it from height h, okay? And you do this for lots of different clips, and you notice that when you plot the time it takes to fall versus height, you see the square root relationship. So this is, t is the time it takes to hit the ground, and this goes as square root of h, okay? And you think this is kind of cool. It's quite regular, although at height, when you go really high, you start seeing departures, which I'll come back to in a second. So here's square root of h, starts departing. This is a really bad drawing. And actually, it eventually gets noisy. So this is kind of cool. You have this regularity. So these are the kind of things we observe. So this is an observation. So this is what I mean by behavior. It's pretty much anything you observe in the world. Okay, and physicists are really good at this because they say, okay, this doesn't make sense, but we can write down F equals ma, and F is mg, and we can solve, and we cancel some terms, we get h equals 1/2 gt squared. Okay, we find the square thing, and so we get this relationship t proportional to root h, right? Okay, so there's some kind of important points hidden in here. First of all, out here, we see departures. Okay, so we maybe add in mg minus nu v, so resistance. But then we say, well, where does nu come from? We may think of Navier-Stokes. I mean, it gets kind of complicated. And then mg really comes from, GMM over r squared, which really came from general relativity. really came from an unknown theory. Okay. But the real point is that when we do this, we kind of do it one step at a time. So we start with a simple theory, set of equations that explains our data, and then we see departures, we add something else simple. Maybe we want to know something about this, we learn about general relativity, but we make small steps, okay? Okay, so that's example number one. I'm gonna come back to that in a second. Example number two is, this is a pretty random example. So we have a tree here. Okay, it's high, let's say 100 meters, pretty tall tree. And so the behavior is water gets the roots to the leaves. And again, we want to write down some equations to solve them. This is actually a little more complicated. And basically, a little thermodynamics. And then maybe some other stuff about water. And that kind of explains the behavior. Okay, and I won't go into details. I actually didn't know this. It turns out the water in the leaves evaporates and it sucks the water up through the capillaries, but that's a complete side point. Okay, so these are two examples in which we've gone from equations, here a bunch of equations describing behavior of, in this case, water and gas, to behavior. It's always equations to behavior. It always involves some kind of solution or approximate solution. And the solution is analytic. Simulations don't add to our understanding. Simulations are useful for other reasons, but they don't really add to our understanding. So what are the take-home messages here? Why am I telling you this? So there are actually two take-home messages. So take-home. One is this is hard. Okay, so Newton had to invent calculus to really write down F equals ma. And the other one, which is sort of obvious but really important, is we don't start with fundamental equations. Okay, so we always, in some sense, we start with approximate equations. Okay, so if you think about it, so down here at the very bottom is true equations describing the universe. You have no idea what they are, but we do have some pretty low-level stuff like standard model. Plus GR, general relativity. But we don't start there, right? We go up, 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 up. And in fact, when describing the tree problem, the water problem, we basically started with thermodynamics. Okay. We actually start with, there's actually something hidden we also started with, and that's, so, you know, this is tree problem. Actually, these are the equations. We actually started with thermodynamics. We also started with something that also wasn't first principles, is that we assumed trees existed. Okay, so we didn't derive trees. We started with the existence of trees, thermodynamics, and water. Okay, and this is, and so, but so this is sort of this one is a point that's going to come up. But the really important part, as we started here. We didn't start down here. Okay, and the reason we understand so much about the world is we can make these small steps. You have some phenomenological equations that explain phenomena, and explain them. It's really some analytic treatment of going from thermodynamics to water. Okay, so now let's talk about the brain. So the brain, neuroscience, let's switch actual neuroscience. Neuroscience is just like any other science. The behavior, we try to explain some behavior, and that's basically everything we do. So it's a bit complicated. I'll say some more about that. And the equations are really, they're differential equations. We actually know what they are, and I'm going to tell you what they are. Describing single neurons and synapses. And then also sort of initial conditions, which in this case is initial architecture. And think, which neurons are connected to which. So this is what we're given. These are the equations. And then we have to explain behavior. Okay, that's the goal of neuroscience. And one of the reasons we've made, or possibly the reason we've made absolutely no progress whatsoever, everybody yells at me for saying we made no progress, but and we made a little bit, is that we're having a hard time finding some intermediate level, sort of the effective thermodynamics. And there's actually, I think, a very, very good reason for that, which I'm going to talk about in a little while. So that's the background. So if you really want to understand neuroscience, the two things you have to understand is the equations that are used and the behavior we're trying to describe. And I'm going to talk about both of those right now. I'm going to switch to slides. Hopefully I'll be able to keep. So I talk fast even when I'm have a whiteboard. I can go even faster with slides. Some of this material will be kind of dense. So you should definitely interrupt me. This should be as informal as possible. Okay. So I'm going to start with what behavior we're trying to explain. And unfortunately, it's pretty much everything. But I'll give you a few examples. So here's a red line and two right angle. And the question is, which one is larger? Which is something you guys would guess on. They're pretty close, so you'll have a hard time getting it right, but you could guess. And this is the kind of problem that's been sort of the mainstay of systems neuroscience for about 5 decades. Okay, neuroscientists love asking animals things like this while recording from neurons. Later on, I'll tell you why I think that's a horribly bad idea, but it's really, really common. But real life things are harder, and I personally am interested in the really hard problems that humans do, the problems that humans are good at, but computers are really bad at. So for instance, here's a kid, tell me a story. This is something you could easily do, right? So we know it's a kid on a bike, it's an urban environment, bad parents, summertime probably. You can make a whole list, right? You might even recognize, well, you probably wouldn't recognize, yes, you might recognize that street. But you can do this really easily, right? You can go from an image to a story. Here's another one. Your significant other who you haven't seen for several weeks is on the other side of a ravine filled with hungry alligators. You jump or take the long way around. Something you guys can solve really easily. Here's another one. You come across some money hanging from a tree. What do? Well, it's pretty easy. You move the rock over, you put the chair on top, you climb up and get the money. And this is something you can do if I change the color of the chair, put it upside down or behind the rock. It has no effect on your ability to solve this problem. And you might even say, this is too good to be true and walk away. And you get other things, right? You watch a movie, then explain the plot to your friends. I show a few pictures. and you build an internal model of something about neuroscience. And this is really my goal. So if I do a good job giving a talk, you'll do that. If I do a bad job, you won't. But this is really my goal. I want you guys to have some kind of model of neuroscience, or more or less the same one I have when this is all over. Okay, but these are the tasks I'm interested in understanding how humans do. Okay, the one of which angle is bigger, your iPhone can do that. But things like this, you watch a movie and explain the plot, or make sense out of a lecture I give. Those are things right now that only humans can do. So what's the point? The point is the brain is complicated stuff, and I'm going to quantify this a little bit. I'm going to quantify it more later on. But if we think about it like this, the brain gets sensory input and produces motor output. We'll call it S&M. And the motor output at a particular time is a function of not just the current sensor input, but sensor input into the past as well. And that's really what makes it hard to understand how the brain works. Everything the brain does in short time scale, we can kind of mimic. It's this long time scale that really, really makes it hard. Okay, and this function, which is a mapping from sensory input to output, is really complicated. So how complicated? Just something like images. So an image consists of about 1 million pixels. Images are not still, they're definitely moving. You get about 1 million bits per second. Your motor output, you have about 600 muscles to drive. And again, this is the hard part. Motor output typically depends on sensor input that occurred decades ago. So for you to make sense of my lectures, first of all, you had to learn language. Then you had to take a bunch of math classes and science classes. And then you had to become good at turning lectures into an internal model. This is not something that, this is not an instantaneous input-output function. This is a really, really hard problem. And the goal of neuroscience is to explain this behavior in terms of low-level equations. Okay, this is a fundamentally daunting task. So this is a behavior. And we're going to switch gears and talk about the low-level equations. That's easy because we mainly know what they are. In fact, we can write down a pretty compact set of equations that describes what's going on in the brain, which is nice. But on the other hand, we can write down F equals ma in quantum mechanics, but we still can't predict trees. So just having the equations doesn't necessarily, well, it's a start, but there's still a lot of stuff, long ways to go. So 2 parts of this. One is the equation describing neural and synaptic dynamics, and the other is neural anatomy, think initial conditions. I'm actually going to start with the second one, because it's kind of nice to have a big picture view of the brain before we start thinking about the lower level equations. This is biology. So biology is, if you hang around biology, it's kind of frustrating. It's nothing like physics. Every fact I know has an exception. It's super irritating. And every single process in the brain, including spike generation, which is relatively simple, has lots of bells and whistles, and nobody has any idea whether they're important. Biology is just plain complicated. I'm going to actually sort of ignore all the details and err on the side of simplicity. And it's simply not known whether and to what extent that's a good idea. But as a first pass, I think it's pretty reasonable. Right? I may or may not be throwing out the baby with the bathwater. Okay. Here's a big picture view of the brain. I've been in neuroscience for 25 years and I still know nothing about neuroanatomy. I finally remembered that there's a frontal lobe that's in the front. And basically most of vision is in the back, and there's all sorts of stuff in between. And the take-home point really is the brain's divided into different areas. It's not one big blob that does stuff. It's really divided into areas, which is useful. If one big blob would be a nightmare. So any other question? Okay. So you can take this cortex is all these ridges and things. Your cortex, you can unfold it. It's about 30 centimeters in radius. If you unfold it, it's got 6 layers. Nobody knows why. About half a centimeter thick. So this blue thing up here is cortex. The red thing is everything else, which I tend not to focus on in my research. It's where
motions reward homeostasis, all the stuff that makes your brain actually work. So for humans, the cortex is super important. For mice, you can take out their cortex and you barely notice behavioral. It's kind of crazy. Cortex is sort of recent. And the reason I think I like it, or maybe theorists like it, is it's pretty regular. There sort of seems like this hope of understanding it. Okay, let's take a closer look. That's a cubic millimeter, about 10 to the minus 3 grams. It's got 50,000 neurons. Each neuron makes about 1,000 connections. That's 50 million connections in this cubic millimeter, 4 kilometers of axons. So it's what sort of we call job security. You should compare that to a transistor, a square millimeter of CPU. It's got 1 million transistors. So the numbers are actually higher, but only 2 connections transistor. I think they're actually 3. I'm not sure if I wrote that. 2 million connections and about 2 meters of wire. A whole brain, or whole human brain, has 100 billion neurons, 100 trillion connections, and 8 million kilometers of wire, which goes to the moon and back. At least once. I think only once. Whereas the whole CPU, I mean, GPUs now basically have more components. Far fewer connections, but far less wire. And so there's something fundamentally different about the way the brain works versus the way the computers work. Although with modern computing power, we can really simulate a brain on any computer. So that's those are numbers you should kind of approximately remember. And really the thing to remember is there are a lot of connections on a lot of wires. I mean, it's actually very sparse connectivity. You have 100 billion neurons and each neuron connects to only 1,000. Still a lot of connections. Smaller picture view of the brain. So this is an electron micrograph. I cannot read this thing. There are about 10 billion cubes of this size in your brain. And this is really this structure. There's all sorts of stuff going on in these very micro levels. I find this particular picture really, really depressing. How in the hell are we ever going to figure this out? Of course, you figure things out by abstracting things away, but this is really what goes on in your brain. It's a miracle it works. So these are, I'm sure some of these are synapses. A neuron is about maybe 20 microns or so across. Okay, maybe a picture just the right size. So your brain is full of neurons. So the picture of a neuron, it's not, it's not quite the scale because, okay, so this is a fact. These are dendrites, that's a soma, that's an axon. For some reason, when I first learned about neuroscience, I could not keep any of those straight. Presumably you guys are smarter. And just for size, neurons are around 20 microns, 10, 20, 30. The genetic tree is about 50 times bigger. It's up to 1 millimeter. It's got to span. So remember, the cortex is sort of this layered structure. It's got to span multiple layers. And the axons are even longer. They're up to a meter. And that's because your toes have to talk to your brain, or talk to your spinal cord at least. So it's a little tiny cell body, and it's really goes a long ways. These are pictures of of neurons. This one's from Ramon Cajal, sort of a famous neuroscientist in around 1900. This is a more recent picture, which isn't nearly as nice, but you can sort of see the cell bodies lives down here, and these are dendritic trees. They're not showing axons. Okay, so they go up a long way, and they can make lots of connections. So what's interesting about neurons is that they spike. They generate voltage. If you stuck an electrode in a neuron and recorded, you'd see the memory potential wandering around. Every now it goes about a threshold, generates a spike, goes up to about plus 20 millivolts, and then back down again. Wanders around. And when a spike occurs, this travels down the axons, and then affects other neurons. So we think of those as wires, and this is your brain, except there are more neurons. All it is a bunch of neurons sending out spikes and receiving spikes, and just a bunch of spikes rattling around in your brain. There's no central controller, there's no central processor, there's no clock, there are just spikes. And so how do they communicate? We can take a closer look at a synapse, which is where basically this is an axon, the blue, and the purple is a dendrite. If there's a connection there when the spike arrives, this current flows into the soma, There's my mouse. Current flows into the soma. You can see that here, and that causes a spike. If there's enough current flows in, the neuron spikes. And we're going to talk, well, we're not going to talk about spike generation, but I'll briefly mention. And so these are the kind of things you have to remember. If neurons turn out to be either excitatory or inhibitory, and they're one or the other, which is kind of crazy. But if neuron J spikes and it's excitatory, the memory potential you'll see on the postsynaptic neuron I, it goes up. On the order of half a millivolt, maybe less, sometimes more. That's called EPSP, excitatory postsynaptic potential. On the other hand, if neuron J is inhibitory, you see this decrease. And again, the range it can be 0.1 to, can't be much more than about a millivolt. But so you see this small deflection. And just put this in scale, sort of this, well, the range down here is about minus 50 is where the threshold is, and then it sort of ranges between minus 50 and minus 65. So this range here is about 15 millivolts. So 20 or 30 of these EPSPs all at the same time is enough to make a neuron spike. The amplitude is, it would call it WIJ, it's going to come up again, and that's changes with learning. And that we believe is where all the actions happen. The change in these weights is really what allows the network to do interesting things. So I'm actually going to write down a set of equations. It's called linear integrate and fire neuron. So this is memory potential on neuron I. If you ignore this term for a second, and basically what it says is, okay, I'm going to head toward rest with time constant tau, which is about 10 milliseconds. And so really what we're describing is not the picture with spikes, but something called subthreshold integration. It's just a passive integration of past input, and it causes this to wander around. So this equation by itself doesn't do anything, but what we can do is just, 10 milliseconds I said, is add on, just say arbitrarily when memory potential reaches threshold, which is about minus 50 millivolts, a spike is emitted and it resets to minus 65. And because it's 1 millisecond so short compared to other time constants, this is called linear integrate and fire neuron. I'm showing the linear integrate part. This is not really what neurons do, but it's not a bad approximation. There are other approximations, but it has the right, it's got the right general framework. And this G is basically caused by presynaptic spikes. So every neuron J spikes, this jumps up and then decays, jumps up and decays. And if W is positive, that causes the voltage to go up. W is negative, that causes the voltage to go down. And there are much more sophisticated versions of neurons, but this is not a bad model. That's about 5 milliseconds. Now, each neuron, as I said, makes about 1,000 connections. That means there are about 1,000 non-zero terms in the sum out of 10 to the 11. And we can throw in learning. And so learning, we kind of know what it looks like. The weight which connects neuron I, J to neuron I, changes with learning. So typically V sub J is really the spikes on neuron J. If the presynaptic neuron spikes, there's some global error signal which we don't understand at all. We mainly don't understand. And so this function F, so people measure this, and you can measure this in the lab. And they're about, you get something that's kind of consistent, but it's very, very different for very different neurons and different synapses and in different conditions. And so we think the tau is much greater than tau. We sort of think learning as going on. It's kind of a slow process, although you can learn things very quickly, obviously. So presuming you remember at least some of what I said. So this equation we understand very well. We can write down a much more sophisticated version of this, which is very, very accurate. This equation, we have lots of candidate functions Fij, but for sure we do not know the right one, in the sense that we don't know an Fij that's going to make this network actually learn something interesting. So we're missing something here, but we kind of know more or less what's going to happen. So this is a complete set of equations describing the brain, right? So W is a matrix. It's size 10 to the 11th by 10 to the 11th. It's very sparse. Each neuron connects about 10 to the third others. We don't know exactly what, I can't tell you exactly what this weight matrix is, but we know a lot about it. We know about connection to different layers, which area connects to which, local versus global. So this is, we kind of have a very good handle on the fundamental equation describing the brain. What we don't understand, what we haven't been able to establish, is some analytical link between these equations and behavior. And it's not a big surprise given how complicated behavior is and how simple the equations are. So we're fundamentally missing. Okay, so to summarize, this is your brain. It's got 10 and 11 neurons. So the red are excitatory neurons. They make about 1,000 connections. For excitatory neurons, these excitatory, I should put that up there. About 90% of the connections are short range. They're local within a few millimeters. The other 10% are long range. Different parts of the brain communicate with each other. And they constitute about 80% of the neurons in your brain. Inhibitory neurons are about 20%. And they're almost exclusively local in the cortex. In other areas, they can be longer range. Cortex are almost exclusively local, meaning a few millimeters, maybe a centimeter. So what do you need to remember? When a neuron spikes, that can cause a small change in the voltage of about 1,000 target neurons. Now I emphasize can because it's this really weird feature, okay? So excitatory neuron, the voltage goes up on about half of its 1,000 target neurons. The other half, absolutely nothing happens. It's called failures. And it's a different half on every spike. So just quite a random half the target neurons fire or get input. Complete mystery why this happens. Same thing with inhibitory neurons. Inhibitory neuron spikes, the voltage goes down on about half its 1,000 target neurons. On the other half, nothing happens. It's a different half on every spike, okay? But really kind of should forget about that and just think excitatory neuron spikes, voltage goes up on its target neurons. Inhibitory neuron spikes, voltage goes down. And it's a pretty good summary of thousands of tens of thousands of person years of work. So it's your brain at the microscopic level. There's lots and lots of structure at the microscopic level. So we kind of divide the brain, at least I divide the brain into three parts. There's sensory processing, which is the input. Motor processing is the output, and in between is deciding what to do. I'm going to draw another picture of that later on. And then you have memory in there. We have no idea what it's for or how it works or where it is, but we do remember things. There are lots of visual areas, lots of auditory areas, lots of motor areas, lots of areas for action selection. All this has to be learned. But as far as the structure goes, it's kind of well known. In fact, there are about 600 different areas, at least in the mouse. So I'm going to actually say, I think we're doing okay on time. I'm going to zip through biophysics just because it's the only thing we know a lot about, and it's actually super cool. I'll zip through biophysics, finish off these lectures, and then we'll take a short break, and then I'll go back to the whiteboard. Okay. So this is, I don't know about you guys, but when I took biology as an undergrad, I hated it. Mainly because I didn't remember a bunch of stuff, but it turns out it's the most cool thing in the world. Biological organisms operate by regulating what goes in and out of cells. It's just a bizarre process. So here's a cell with a bunch of things inside it. This is going to be ions. They have channels and stuff flows out. And when stuff goes out and influences other cells, it's actually kind of a crazy system. So here are two cells. Let's say a channel opens. Ions come out in this case. In the case of neurons, they diffuse across, they open the channel, something happens, and then other things come out. And if the cells have this shape, communication is very local. And they can do complicated things, but not that complicated. And the reason neurons are interesting is because they're very long range. So now a single cell can influence not just local cells, but cells that are, many cells that are, can influence about 1,000 cells, some of them many, many centimeters or even meters away. And so for neurons, the red triangles are charged ions, sodium chloride, potassium, calcium, magnesium, not to mention the inside, but on the outside. There are lots of channels and there are ion pumps to keep there's an ion imbalance. So neurons, the amount of, let's say, sodium on the inside and the outside is very different. So this is a story, and what we really care about is really the relationship between voltage and current in a neuron. We eventually want to write down a differential equation. So we all know V equals IR, which you write I equals 1 over R times V because current is sort of fundamental. And one of our inverse resistance is conductance, so we write I equals GV. And this is clearly wrong. And the reason it's wrong is because there's an ion imbalance. You have ion pumps that will put a lot of one kind of ion on the inside and not much on the outside. And just from diffusion, that's going to cause a current. So we can easily fix that. We just cross that off and just replace it by G times V minus E. So E is called the reversal potential. It's the voltage needed to keep any current from flowing. So if these were positive ions here, we need a big negative voltage in the inside. So the voltage would have to be much smaller on the inside to keep it from flowing. So in this case, if these were positive, we'd have a negative reversal potential. If these were negative, we have a positive reversal potential. We need more voltage on the inside than the outside. So this is pretty much it for current. It's pretty much universal for cells, okay? And current flow causes a change in voltage, right? So C dV dt is basically Q equals CV in disguise. I is the outward current relative to the inside of the cell. So what this says is, as current flows out of the cell, the voltage goes down, and the amount that goes down is some constant, which is capacitance. Okay, and so I we can replace by D times V minus D. So this would be for the whole story. This would be kind of like an RC circuit. But there are lots of different kind of channels. So different kind of channels allow different ions to flow. And we get sort of, think of this as resistors in parallel. So we have the sum of the conductances times voltage, and different ions may have different reversal potential. So this is super whirlwind tour. Different channels allow different ions through the cell membrane. So this is basically, so far has been all physics. The biology comes in because the conductances, so we used to, in resistors, we used to sort of constant conductances. Okay, it's got a resistance and that's it. Transistors aren't like that, and transistors are much more interesting. Same thing in biology. These conductances can change. And if you could fill in a ... everywhere, you would have a complete description of neurons, axons, dendrites, and synapses. And we've mainly done that for neurons, axons, and dendrites, but not for synapses. I'm going to give you two examples, action potentials, which is something like this. So it turns out there's a lot more sodium on the outside of a cell than the inside. And the sodium conductors is voltage dependent. Okay, so when the voltage is low, the sodium conductance is low and not much happens. Okay, the sodium kind of, trickles in, the pumps push it out. If the voltage ever goes up, remember we saw a threshold about minus 50 millivolts. Somewhere around minus 50 millivolts, the sodium conductance goes through the roof, sodium rushes in, and the voltage goes way up. At really high voltage, it shuts down again. And so this is basically what causes an action potential. Right here, sodium channels open, up on the top, sodium channels close. Other things happen, and then that's it. So the voltage goes down. So a spike is really caused by sodium conductance. First of all, ion pumps put a bunch of sodium on the outside, and the sodium conductance that's voltage dependent. Okay, and this gives rise to Hodge complexity equations and a Nobel Prize. Those were the good old days when it's easy to get a Nobel Prize. Now it's a lot harder. Actually, not that much harder. Okay, the other synaptic transmission, and this is just the same idea, except G is now going to depend on something else. So this is kind of a picture you should have in your brain. This is an axon, comes from the soma. This thing here is a presynaptic terminal. So I drew that picture earlier. The axon was blue and the dendrites were purple. The same thing, but a blown up picture. So the axons branch and then synapse often on presynaptic terminals. It's a postsynaptic terminal, that's a spine, and that's a dendrite, and that's the soma. And it's kind of insane the way neurons communicate. So what happens is a spike. So first of all, this axon is filled with little vesicles filled with neurotransmitter. What happens is a spike comes down, opens calcium channels. I mean, it's sort of cool in the sense that it's just a kludge. The calcium channel causes all sorts of intercellular things to happen, which I don't understand at all. And that eventually causes one of these vesicles to fuse and release neurotransmitter, which binds to the cell, opens a channel, and current flows. Okay, so it's a really, really complicated process. You have 100 trillion of these in your brain. 100 trillion synapses. Neurons are firing at about probably less than a hertz. So basically, each of these synapses is releasing neurotransmitter, except when it doesn't, several times a second. So it's got 100 trillion of these events a second. And it just works for decades. It's a miracle. It's a really strange system. But the point is that the conductance is just now, instead of voltage dependent, it's a function of concentration. Okay, so as the concentration goes up, the synaptic conductance goes up and current flows. But it's the same idea, right? It's always V minus E something, and then some conductance changes. Okay, summary of all of biophysics is a fundamental equation, which is this. The change in voltage is determined by conductance and then reversal potential. This is V equals IR, souped up by the fact that there's an ion imbalance. That's reversal potential, that's conductance. And you get almost an infinite variety of behavior through the conductance GX, which can depend on voltage. It can depend on, main things it depends on are voltage and concentration of some neuromodulator, neurotransmitter. In periphery, it can depend on all sorts of things. Your eyes, photons can change the conductance. In your hairs, mechanical in your ear, mechanical hair cells change the conductance. So this is basically what biology uses. Okay, what you should remember. So the first part of behavior, which is the mapping from sensory input to motor output, is complicated. And it's really insanely complicated. I mean, the behavior we do is really, really hard to even describe. The brain is divided into different areas. We saw a picture like that. There are about 600 of these areas. And these are just visual areas. This is how, not only which they are, but how they're connected. This is really a subset. It's kind of an old slide. Each neuron makes about 1,000 connections. We have no idea as to how important that number 1,000 is. It's sort of consistent across mammalian species. Everything from mouse to humans to elephants. So your brain at microscopic level, excitatory neurons. You mainly got excitatory neurons, who mainly make short-range connections, but sometimes make long-range ones. There are lots of neurons in your brain. There are 10 to 11th neurons, and that comes with 10 to 14th connections. These are really, really large numbers, and 8 million kilometers of axons. So these are the kind of things you should have at the back of your mind. The first I can always remember. The third one I have a problem with. Low-level equations aren't that complicated, right? Your equations for voltage. The true ones are a little bit more complicated than this, especially if you throw in dendrites. And there are learning rules. The weights get updated. We don't quite know what the learning rules are, but we know they exist. Okay. So I'm going to end with a current view of what the brain does. And this has sort of evolved and developed over sort of decades of thinking about it. And it's not that complicated in a way. So here's our brain. You get sensory input, sounds, images, smells, and so on. And the first thing that happens is short-term sensory processing. And so we're very visual creatures. If you walk into a room, within about a second, you know all sorts of stuff. You know who's in there, what's in there, how it relates. And this happens really, really fast. If you hear things, if you smell things, you immediately know all sorts of stuff. And one of the things we do is construct a model of the world sort of on long time scales. The reason you can do this short-term sensory processing fast is you know stuff, right? You know what a chair is, you know what a person is, you know what a building is, you know all sorts. But given that sort of the short-term sensory processing and our model of the world, you choose an action, you execute a motor command, and then that affects the outside world. So this is it. This is what the brain, pretty much what it does. Now there are sort of feedback loops, right? Having a model of the world, it helps you interpret it. If you execute a motor command, you better tell your sensory system, or it'll think that, you know, if you move your eyes and you haven't told the world about it, it'll think the sensory system will think the world's moving. Sensory, actually, when you execute a motor command, it's not open loop. You know, you can actually affect what motor command you execute. And there's all sorts of stuff that goes on. And so the crazy thing is, in some ways, we kind of know how it does all this. So we can build deep feed-forward networks to do sensory processing really, really well. They don't quite hit human level performance in some ways. In some ways they're better, in some ways they're worse, but they do pretty well. choosing an act, you execute a motor command, given that we want to do something, we want to reach out and grab something, the current neural networks are now doing a pretty good job. I mean, robotics is not solved, but it's going to be. And it's basically, it's just driving about 600 muscles and generating the right trajectories. Hard problem, but not insanely hard. So choosing an action is surprisingly hard. But DeepRL can now solve these in specific cases. It crack Go. You can do many tasks much, much better than you can. And so these three things, we kind of know in some ways how the brain does it. Now, when I say no, what we don't know is how deep feed-forward networks work, how deep RL works, or how recurrent networks work. We know they work, we can train them, but we have no idea how they work. Something I'll return to. And then this last part, we have no clue whatsoever. You crack that, you crack AI, and it's the end of humanity. But we this is the brain, right? It just does some hard tasks, sensory processing, choose an action, execute a motor command, and that's it. And the reason that we do that so well is basically all this past sensory input has given us a really, really good model of the world. We can generalize in new situations. We know what to do. We know what to ignore and what not to ignore. And exactly how that's done, we have no idea. Okay, so let's take a break. I'm going to go back to the whiteboard. And so the last thing I want to talk about is okay, it's complicated. We know the equations. We know what behavior we're trying to get at. How do we go back at figuring out how the brain works? Okay, I'm going to turn this off. So let's take about a 5-minute break, and you guys can feel free to ask questions. I'll be back in about 1 minute.
Okay.
Yeah, I'm here. Hi, Peter. Should we continue? Wait.
Whatever. Students have been sat through 4 hours of lectures today, right? Probably burned out. How much time do I have?
So initially we had scheduled until 6 here, which would be 5 for you, but
Okay.
maybe the break has gone a little bit longer than intended. So
Well, I probably have. Who knows how much I have? I probably have. Well, I don't know. Well, last part is kind of fluff, so it's pretty easy.
okay, so let me get the people in here, and we'll continue. Here they're coming in. We actually have this kind of room microphone that we were hoping to. It was working yesterday, and today, of course, when we began, it didn't work. We're hoping we'll get it working tomorrow again.
I mean, students can also type in
Because it's much better for questions and so on. Otherwise, people have to shout or come to the front, and that's always an additional.
They can also type in the chat, right?
Yeah, that's true. But the people that are here in person cannot really chat. But
they can't do that. That's a good point. Sorry. Who are the students? Are they mainly from Norway?
No, we have quite a mix of people from other places as well.
Cool.
Yeah, so it says he's from Norway.
Who's from Norway? Yes, sir.
Apparently so. Okay. Yeah, we're all ready to continue if you want to.
Okay. Okay, so let me share screen. Voila. So what do we have so far? We have that we kind of know the underlying equations describing neurons. And we have some idea of how synapses evolve in time. We know a lot about behavior, mainly because behavior is about us. And on top of that, there have been tons of experimental people do experimental psychology all the time. So we know really a lot about the way animals behave, especially in sort of the way humans behave. And then we want to link it together. So let me start with sort of some deceptively simple looking equations. So remember, motor output is some function of the sensor input from when you were born to the current time. It's all M of t. So this is motor output. Okay. And on top of that, then we also have on the other side, dv I dt equals and dw I j dt. So these are the equations that we have, and we want to somehow link them over here. So the first thing we do is, so this writing motor as a function of all past input is really, that could be almost anything, but we can do a little bit better than that, because sort of the standard view is that the motor output at a given time I'll use a different function. It depends on S of t and weights. So this is current, I'll put current in quotes, sensory input. The reason it's in quotes is because, it could be the last tens or even hundreds of milliseconds. Or maybe in some cases seconds, and these are the weights. The things that connect neurons from each other. And the weights are another function of, the weights are actually a function of the past input. Are a function of S 0 to t. Okay, and they actually are updated locally in time. And so most of systems neuroscience has focused on this. Well, when I say systems neuroscience, I mean the kind of neurosciences that stick electrodes in animals' heads and try to figure out the relationship between these equations and behavior. And the reason for that is that animals, to do anything, I guess you may see something about this. If you want to do experiments, you have to do lots and lots of different trials. You need lots and lots of spikes. And so the animals, when they learn, they're non-stationary, right? As this W evolves in time, this input-output function changes, and characterizing it gets kind of hard. So people focus on, they'll train an animal, and then after they train it, they'll try to figure out, well, how's the transformation done? Okay, it's kind of important point. Kind of in neuroscience, so systems neuroscience, I mean, not certainly 100% of, so much of systems neuroscience, focuses on behavior after learning. Okay. They think the weight's being frozen. They say, how is it done? How is how is this sensory input, this current sensory input translated to motor output? And it seems like an easier question. I'm going to argue actually it's probably a bad idea, but it's almost by definition easier. Okay. Now, of course, we don't have to do this all in one step. Okay. Remember a picture of the brain, there's sensory input, S of t, there are a bunch of boxes, only 4. But motor output actually sits here. And so and so this is going from sensory input all the way to motor output. It's not necessary. It's pretty much overkill. Let's actually focus on something that's also been heavily studied in neuroscience. We're going to focus on and we can think of that as extracting latent variables. So I don't know, in this day and age, maybe everybody knows what latent variables are. I arrived at Gatsby in 2004, and people kept talking about latent variables, and I was too embarrassed to ask. It took me 3 years to figure out what they are. And they're not actually very well defined. So latent variables are the things you actually care about. So if you think of an image, right, worst star in the world. If you think of an image, it's just a bunch of pixels, right? You don't care about the pixels. You care about, this is the house, got a window. So these are latent variables. And so that you basically, a network, you can think of S, we're going to write Z, well, we'll let L for latent, well, Z equals some function of S. So this is sensory input. And those are latents. So this is a step that we believe animals and humans do. It's almost for sure we do that. You can imagine just sort of some network that just takes sensory input and goes straight to motor output. Almost for sure we take the intermediate state of extracting latents, and instead of passing these latents, that's what gets passed around here. Okay, so let's just think of that problem. And we can think of this, in terms of, if it's S for visual scene, this would be object recognition. Okay. And we kind of want to know, so our goal is, so if we're going to focus on sensory processing, the goal is, how do we do it? The goal is kind of easy. How do we go from S to Z? How do we go is really, really poorly defined. But so David Marr, the famous David Marr, is probably especially famous because he died young, had the following kind of very reasonable approach. In fact, this approach used in most other sciences is forget about the brain. So when we say, how do we go from this? We mean, how do neurons do it? Okay. That's really the question we want to ask. So David Marr said, forget about neurons, think about algorithms. Okay. And then once we know the algorithm, that's step one. And step two, once we know the algorithm, think about neural implementation. Okay, it's pretty reasonable, right? Because neurons are a bit of a nightmare. So thinking about algorithms first would seem to make our life easier. And in some cases, that really works pretty well. So a really classic one is decision-making. People spend I'm guessing 100 million dollars worth of research money has been spent on this. So simple decision-making. So in a classic task, you'll see a bunch of moving things, moving dots on a screen that's kind of in random directions. And the question is, are they going right or left? They're moving right or left. Oh, the random dot kinematogram. Super famous. And it's pretty easy to write down an optimal algorithm, right? You just have motion detectors. The motion detector, if you basically look at average motion versus time, it's going to be, it's going to wander around because it's pretty noisy. Eventually it's there, and then, this choose. Right. So that's an algorithm. And the algorithm you can talk about without referring to, this is sort of evidence. Okay. This is exactly the coin flip problem. I've had biased coins in my pocket. One's 80% heads, one's 80% tails. I pull one out, I start flipping it, and you ask you to tell me whether it's heads or tails. You would wait till you see, a bunch of heads in a row or a bunch of tails in a row before you make a decision. That's exactly what's happening here. But this sort of algorithm doesn't even mention neurons. Okay, so it identifies the fundamental features here, which is you have to look at the evidence, you have to integrate it because instantaneous evidence is super noisy, and when you see enough of it, you make a decision. As a classic case, we can write down an algorithm, and then because we now actually know a lot about neurons, we can build networks of neurons to actually do exactly this. We can look in the brain and we can ask for, let's see if we can find areas where we see evidence for this integration, see evidence for this instantaneous motion. We actually do, except it turns out if you ablate the area that does the integration, the brain still does it just fine. So we don't really know what's going on. But in simple tasks, this idea of writing down the algorithm and then turning into neural implementation probably works in a way, in the sense that we can do it, we can write down the algorithm, we can, and neural implementation kind of matches what we see in the brain. It's because the brain's big, everything matches. But let's ask if this is in general a good idea. So we're going to go back to our just simple extracting latent variables, z equals f of s. So we're going to call this a vector because think of an image, or even sound. This is a complicated, sensory input is complicated. And we can ask how complicated is this function? So now I'm going to make a kind of a strong claim, and that is, if it's too complicated, it's not human understandable. The mapping from sensory input to latent variables, we can understand it, and we actually kind of don't even want to. Okay, so that's a really strong statement. If it's too complicated, and any function that's really too complicated is not even understandable. So what do I mean by complicated? So I'm going to measure complexity, C, as log, we'll do log to the base 10 so we can easily interpret it, of flops, floating point operations per input dimension per time, say. Okay. So these are floating point operations. Okay. And if this were images, for images, this is pixels. Okay. So if I present an image and I want to recognize it, if it takes, so see, let's say C equals 4. That implies it takes 10,000 floating point operations per pixel. Recognize it. Okay. And that implies it is too complicated and not even worth understanding. So I should be getting a little bit of pushback from this because it's not really sort of fundamentally true. So for instance, let's say you want to factor a coprime. I give you a number that's 10,000 digits. It's a product of 2 primes. And I want you to tell me which one it is. It's going to take you a lot of flops. But you deeply understand the computation. Okay? So just because it takes a lot of computing power doesn't mean the function is not understandable. So this is not something provable. This is kind of like, this is currently opinion. I need to actually sit down and read the literature because computer scientists worry about this all the time. In some sense, we really care about Kolmogorov complexity, which is the shortest program that you can write to do some task. And initially, I don't know if you guys know this story, in the 60s, they hired some researcher, postdoc, grad student, I'm not really sure. And the summer project was going to be to understand computer vision. So really understand the transformation from sensory input to the object that was in the picture. And it's going to be easy, you know, because vision's really easy. Look at it, you know what it is, right? It's got features. That was 1960. It's now 60 years later, and nobody has found a shortcut. The only way we can recognize images is through a deep network at it and perform on the order of 10,000 floating point operations per pixel. So it's kind of interesting to count complexity. So let's say Z equals W S summon I of W I S. So N, so number of pixels equals N, number of operations equals 2N. So we have to add up, we have N multiplications. We have N multiplications and then N addition is actually N minus 1. So, flops, so log to the base 10 of flops per pixel equals log to the base 10 of 2 equals small, I think it's 0.3, something like that. So yeah, so linear transformations we understand. What about a deep network, which will actually recognize objects? Say we have N layer. Let's just say everything is N. So if N neurons in each network. So if you look at each layer, right? So number of flops is equal to what's 2N, because the output is it's some function of the sum on J of Wi J X J. So for each unit I, we have 2N operations. So number of flops is 2N times N. And that's because there are N units in this layer. Okay. In the next layer, it's plus 2N plus 2 plus 2n times n. It's basically, it's a length, it's 2 times this times n squared. That's number of flops. We divide that by n, number of pixels, and we get 2ln equals number of parameters. Number of units. Okay, so the complexity of a deep network is basically is log. So complexity C for deep network equals log of 2 times number of units. Okay, and they're easily 10,000 in here. It can be even larger. Okay, so we have exactly one, I don't know if you call them algorithm. We know of exactly one way to do object recognition, and it's basically, its complexity is really, really big. It's on the order of 4 to 5, maybe 3 for smaller networks. You can make it smaller, right? But even in complexity of 2, even if you have to do 100 floating point operations per pixel, forget it. It's just not, it's not even something we want to understand, okay? If we're really implementing complicated function, it's just a complicated function. Trying to understand how it's done. And remember, sort of in the beginning, this is what we want to do, right? We want to, in terms of these equations, oops. So we're going to look at these equations, explain how this transformation is done, or this transformation is done. Doesn't make sense. Okay? That's a very, very strong statement that might be wrong. So I'm going to go, I'll end pretty soon because it's getting late. So we can also think of the brain divided into areas. These areas are connected. And we can think of the input-output of a particular area, right? So, y equals f of x. And we can ask the same complexity. Typically, number of pixels becomes number of neurons. Which in the brain is large, right? Often 10 to the 6 to 7 to 8. It's big. But inside this network is recurrent circuitry. And you can sort of add up flops the same way we did before, and we get on the order of log number of units in the network. Which generally dwarfs the number of inputs. Number of units in the network divided by number of units. Okay, and that's greater than 2 or so. It's big. So that means individual areas in the brain are also not really understandable. The full, and this again is kind of an assumption. If those areas are actually doing something simple, something reducible to a few flops per input, then yeah, you can understand it. If they're not, then they're not human understandable. Okay, So why do neuroscience? So suppose we spend a lot of time looking at simple tasks. Are we going to learn anything? So a simple task, if you look at simple tasks, what it is z equals f of s naught plus delta s. Well, this is low d. Okay. And so what experimentalists do is fix everything, and they vary one thing. So the random dot kinematogram, everything was fixed except the dots on the screen. The animal didn't have to worry about anything else except the dots on the screen. And of course, we can come up with an algorithm. But if this is fundamentally complicated transformation, so S naught goes to S1, and we know nothing. But we know nothing given we know what happens. So this is an important point. Let's say we buy that if for complex transformations, they're not human understandable. This is effectively what it means is if you understand something, look at small perturbations around some input, because it's not human understandable, it fundamentally takes 10,000 floating point operation per pixel, then we're simply not going to learn anything about anything else. Okay, so I'm going to end right there. And this is, I'm going to end with my current view of all this. So let's sort of summarize. Transformations brain does. They're not human understandable. Okay, so this, of course, is opinion. But let's pretend like it's true. So what does that mean? Simple tasks means simple enough that we can understand them. Tell us very little about how the brain works. And that would seem, a simple task, these two statements would seem to be incredibly depressing. They're not. We just have to change which problems we work on. It's like in the 80s when they discovered chaos and they realized the differential equations were often sensitive to initial conditions. So you don't try to solve them. You ask different questions. And in my questions, all interesting questions, and I'm guessing this is a theme you're going to see throughout this course, given who's speaking, are about learning. It makes very little sense to look at a network after it's learned. The learning trajectories are interesting. And learning actually is a sort of a small number of parameters, right? You can have one learning rule. You can ask, given an architecture and a learning rule, so this is an interesting and answerable question. And in the end, I think, the picture of how the brain's going to work is going to be, okay, the initial architecture was sufficiently expressive and it had the right learning rule so that it worked. We won't be able to say that much more. And finally, I'm going to leave you for this. You're going to hear lots of talks and you're going to get lots of facts. And you should always ask, skeptical anybody is going to ask anything given I got 0 questions, but you can always ask, so this is, you should always ask that to yourself and the speakers as well. So I'm going to end there. To summarize, so I told you a lot about single neurons, a little bit about the kind of behavior we're trying to explain, and why I think it's hard and in neuroscience generally misguided. Okay, I'm going to save. I'll send you guys all the photos so you can have these slides so you can have them if you want. And I'll stick around if anybody has any questions. You can, I guess the ones in the lecture, it's going to be tough, but I'll be here for a little while.
Thank you very much, Peter.
Sure.
Do we have any questions now, or also from the people online?
I think I saw it in chat.
It was
No
Nicolai's
questions.
thing.
And you know, if you guys have burning questions, you can email me.
Okay.
I guarantee it.
Yeah, we can pick up on Friday. Yeah.
That's true. I'll be back on Friday talking about actual math.
Oh, there's a question here in the back. Can you come here? Sorry, she's coming to the microphone.
It strikes me.
So yes, so the question is about is there a way to unify the simple model you introduced with the biophysical equations? So the answer is, the simple model refers to the decision-making model.
I can give
Is that correct?
the power to speak.
There.
Now,
Well, assume
to be able to speak.
Oh, hello.
Hi.
Hi. Yeah, I was just. It was referring to the simple model you introduced with motor output equals the function, some function with input as sensory input over all time. And I was wondering how you can. Is there any work done in linking that simple model with the biophysical equations of the signal transferring through neural networks, for example?
Yeah. So you're referring to this equation here, the red one?
Yes, correct. Yep.
So the red one is generally thought of, so there's, you have to store information in the past. It's generally believed those are stored in the weights.
Okay.
Synaptic strength between neurons.
Okay.
So, given the weights, so this equation refers to a neural network, a recurrent network. I'm going to talk about those on Friday. And this is the update rule for the weights. So the past is stored in the weights, and then given the weights, you implement some function. Did that answer your question?
Okay. Yeah. Thank you.
But yeah, there's a mismatch of timescales, right? Neurons operate in 10 millisecond timescales, whereas you have to have memory on decades.
Yeah. Would it be correct in thinking that the equation on the left, the simple one, is kind of like an event-driven equation? So the timescales are like between events, whereas the right is kind of like microscopic. It's like as an event happens.
So the right is microscopic. The left is really just a placeholder. I mean, it just tells you, it's just a way of, I don't know, writing this equation may be sort of irritatingly misleading. All it says is, look at, if I want to predict what an organism is going to do now, I have to know its past sensory input for all time.
Okay, got you.
But not compressed, right? I mean, so if I have to know that you're an English speaker, or I know you couldn't respond to me, things like that.
Yeah.
I don't have to know in detail. But you certainly use all past information to set your weights and allow you to respond currently.
Thank you.
Sure.
Okay, so I have a question about the statement that all interesting questions are about learning. I wonder, how would you position experience or like states of being in that framework? Is it that the system has learned the parameters which dictate a certain state of being in response to certain conditions? Or do you have any thoughts on that?
So when you say state of being, are you referring to the fact that, when we get input, it changes our mood and what we think about things rather than actually generating an action? Is that what you're referring to?
Yeah, so you could go from doing nothing to feeling angry or feeling stimulated, or like just not actually solving any explicit problems, but just changes in state of being, let's say.
So, as I said at the beginning of the talk, all interesting questions about neuroscience is we have no idea. So I think, this idea that motor output means actually moving muscles, I guess, is what you really, a lot of what you do is change, you're right, you change your internal state. And that should somehow be reflected. I'm not quite sure where to put that. But certainly you have 100 billion neurons floating around, and your input changes your internal state. And one of the things that changes is how you feel about things. And that's certainly going to be ultimately part of any theory we have. And in fact, it's sort of incorporated to reinforcement learning. So you get punishments, and people have theories about theories of vigor. Why work hard or why not work hard? So the answer I'm guessing was very unsatisfying, mainly because we don't know much. But I think ultimately it's going to have to be taken into account that external input changes your internal state.
Okay, I think we can wrap there for today.
Okay.
Thank you, Peter.